Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In today's video, we're going to take a look at gene expression. What that means is how do we go from gene to protein? Follow along with me as we'll go through each step, and by the end, you'll understand how we go from the DNA, the gene, to the protein, the functional product. DNA is the genetic material. It is inherited by an organism and leads to specific traits by dictating the synthesis of proteins. Proteins are the links between genotype, what we see in the DNA or the gene, and phenotype, what we physically see in that protein. Gene expression is the process by which DNA directs protein synthesis. It includes two stages, transcription and translation. Transcription is the synthesis of RNA using information in DNA. Translation is the synthesis of a polypeptide using information in the mRNA. So transcription is going from DNA to RNA, specifically mRNA, which is the messenger RNA. And translation is taking that information in the mRNA and converting that into a protein. In prokaryotes like bacteria, the translation of mRNA can actually begin before transcription has finished, and this is because bacteria do not have membrane-bound organelles, so they do not have a nucleus. So the DNA and RNA, this all has access to everything that is necessary for both transcription and translation. So bacteria can actually allow for translation of mRNA to happen before transcription has even finished. However, in a eukaryotic cell, because there is a nucleus with a nuclear envelope, this actually separates the processes of transcription and translation. So transcription is going to happen in the nucleus, and translation happens in the cytoplasm where the ribosomes are in order to be able to make protein. So since they happen in two different places, in the eukaryotic cell, they cannot occur at the same time. In this figure, we can see that since bacterial cells do not have a nuclear membrane, its DNA can be transcribed into mRNA, and at the same time, there can be translation occurring of that mRNA. However, in the eukaryotic cell, because of that nuclear envelope that we just talked about, DNA to RNA, or mRNA specifically, is going to happen within the nucleus. So the process of transcription in eukaryotic cells is going to happen in the nucleus, and you can see that mRNA also gets processed. Bacteria do not need this mRNA processing either, which is another reason that transcription and translation can occur simultaneously. So in the eukaryotic cell, DNA will be made into pre-mRNA, which then needs to be processed into mRNA that would be ready to leave the nucleus and attach to a ribosome in the cytoplasm, where translation would be able to occur. We can talk about the central dogma, that is the concept that cells are governed by a cellular chain of command that includes DNA being the hereditary information, being able to be transcribed into RNA, which can then be translated into protein. So it goes from DNA to RNA to protein. How are the instructions for assembling amino acids into proteins encoded in the DNA? There are actually 20 amino acids, but there are only 4 nucleotide bases in DNA. So how many nucleotides correspond to an amino acid? The answer is 3. there are 3 nucleotides needed to code for one amino acid. And this flow of information from gene to protein is based on this triplet code. So the triplet code, again, is that there are 3 nucleotides that will code for one amino acid. The words of a gene are transcribed into complementary, non-overlapping 3 nucleotide words of mRNA. So both the DNA and the RNA or mRNA are both read in codons, which are groups of 3 nucleotides. And then when we move to protein, we're then talking about amino acids. So these words are then translated into chains of amino acids. Okay, so DNA and RNA both contain nucleotides, and those nucleotides are read in groups of 3s called codons. When we move to protein, though, protein is going to be made of amino acids. So for every 3 nucleotides, we are going to be able to code for 1 amino acid. Let's take a look at this figure and start to put some of these things together. So we're going to go from one strand of DNA called the template strand, and from that template strand of DNA, we're going to be able to transcribe mRNA. Now remember, DNA and RNA are both nucleic acids, so they are both made of nucleotides. The only difference is that RNA has uracil instead of DNA having thymine. They do also have different sugars. So when we take that DNA template strand, it will be used to make that mRNA, that piece of mRNA that we're going to need in order to make protein. So that process is called transcription, and that is going to occur in the nucleus. So here is our DNA template strand where we have an A, and the correct pair would be a U or a uracil. We'll have the C in the DNA template strand being paired with that G. So we're going to have a G in our mRNA strand. Again, for the C, we're going to have another G. For the A, we would have a U, another U for the next A U for the next A, and then we have CC, so we have 2 G's, and then GAG, which would be CUC, and then for that last T, we would have an A. Then what's going to happen is that the mRNA is going to be read in 3s, those codons, in order to figure out what amino acid needs to be placed for the protein. So every 3 nucleotides, every codon, the 3 nucleotides equals 1 codon. For every codon, there is 1 amino acid that is going to be placed together to make the protein. Now, translation is going to occur in the cytoplasm on a ribosome, and that is where these codons are going to be read in order to put the correct amino acid, and we'll be talking about how that happens later on. So like I just mentioned, one of the 2 DNA strands is going to be called the template strand, and that template strand is going to be used as the template to provide the correct complementary nucleotides in order to synthesize the mRNA, like I just showed you on the previous figure. The template strand is always the same strand for a given gene. The mRNA molecule that is produced is complementary to that template strand, like I just showed you as I went over that figure. During translation, the mRNA-based triplets, which are called codons, are going to be read in the 5' to 3' direction. Each codon is going to specify the amino acid, which is going to be one of 20. There's 20 different amino acids, and that codon is going to specify the amino acid that is going to be placed at the corresponding position along a polypeptide as the ribosome is synthesizing the protein. All 64 codons were deciphered by the mid-1960s. Of the 64 triplets, 61 code for amino acids, while the other 3 triplets are stop signals to end translation. The genetic code is redundant. What this means is that more than one codon may specify a particular amino acid. But it is not ambiguous, so no codon specifies more than one amino acid. So the way we read these nucleotides in these sets of 3 in these codons is referred to as the reading frame. There is only one reading frame that is correct or encoding the correct sequence of amino acids. And if a frame shift mutation occurs where it moves, the reading frame is shifted, then a defective product could be made, because then we're shifting the way we read the codons. So the codons have to be read a specific way, and if it shifts, it could make a defective product. There are 3 stop codons. They are shown here, where they are UUA, UGA, and UAG. They are all used to terminate translation. So there could be different ones, but whenever these are in the reading frame, they will stop translation. And there is only one start codon, which is AUG, and that is used to start translation. If AUG is not present, then synthesis does not occur. This figure shows what the genetic code looks like and how we can use this to be able to figure out what our mRNA codon is and what amino acid it's coding for. So what we need to do here is we would find our first mRNA base on this side, our second mRNA base on this side, and we would use the final side to find our third base in the codon. So groups of 3 to figure out what amino acid it's coding for. So let's just do an example so that you can hopefully understand how this works. So let's say that our mRNA sequence is AGA. So we look on this one side first, this left-hand side, we find the A. Our next one is G, so it's right here. So we come down to where the A was because we have to be here. So A was the first, G was the second, so we're now in this box. And our last one is A, which is right here, which brings us right here, AGA, and it is the amino acid arginine is what that would code for. So we can figure out what the amino acid would be. So again, mRNA and DNA are both read in triplets called codons. So 3 nucleotides codes for 1 amino acid, and we can find exactly what amino acid is here. What we can also see from this genetic code is that more than one codon can code for the same amino acid. Sometimes even several codons can code for the same amino acid. So that's where we were talking about how the genetic code is redundant, because several codons can code for the same amino acid. However, only one codon, every codon is only going to code for one amino acid. So it's not ambiguous. They're not coding for more than one. Each codon is only coding for one amino acid, and we know what that amino acid is. So we could use this chart to figure out what our amino acids are if we have the mRNA sequence. As I mentioned already, transcription is going to occur in the nucleus of the cell. The synthesis of RNA is going to be catalyzed by RNA polymerase. Now, because we are making RNA from that DNA. The enzyme that is going to be used in this case is RNA polymerase. And RNA polymerase is going to pry the DNA strands apart and join together the RNA nucleotides to synthesize that RNA from the DNA template. RNA synthesis is going to follow the same base pairing rules as DNA, except that uracil is going to substitute for thymine. So we see thymine only in DNA, and we see uracil in RNA. The DNA sequence where RNA polymerase attaches on the DNA is called the promoter. So in order to have transcription start, where we're having that synthesis of RNA from that DNA template, we are going to have the RNA polymerase bind to the promoter on the DNA. Once RNA polymerase binds to the promoter, then it can unwind the DNA and start the synthesis of RNA from the DNA template. The DNA sequence where the RNA polymerase attaches to the DNA is called the promoter. In bacteria, the sequence signaling the end of transcription is called the terminator sequence. The stretch of DNA that is transcribed is called a transcription unit. There are 3 stages of transcription: initiation, where RNA polymerase is going to bind; elongation, where we're going to elongate that RNA throughout the RNA synthesis; and then there's going to be termination, or the ending of transcription. Promoters are going to signal the transcription start point, and they usually extend several dozen nucleotide pairs upstream from the start point to give RNA polymerase a little bit of an area to be able to bind to. Transcription factors are going to help guide the binding of RNA polymerase in the initiation of transcription. The completed assembly of transcription factors and RNA polymerase are going to bound to the promoter is called the transcription initiation complex. A promoter called a Tata box is crucial in forming the initiation complex in eukaryotic cells. We can put all that together here. So the promoter includes a good bit of nucleotides within it. And within the promoter, we have that Tata box that is crucial in forming that initiation complex in eukaryotes. So the promoter is going to be the region where RNA polymerase binds. In order for RNA polymerase to bind, transcription factors also need to bind, and they are going to help guide the RNA polymerase to bind in this area. So now you can see the transcription factors bound first, and then we have RNA polymerase coming to this area as well. So the RNA polymerase plus these transcription factors all on this promoter area is called the transcription initiation complex, because this is what's going to initiate the start of transcription to allow that DNA template to be synthesized into RNA. Once the transcription initiation complex has initiated the beginning of transcription, then elongation can occur. So the RNA polymerase is going to move along the DNA, and as it does, it's going to untwist the double helix about 10 to 20 base pairs at a time. Transcription is going to progress at a rate of about 40 nucleotides per second in eukaryotes. The nucleotides are going to be added at the 3' end of the growing RNA molecule. So in this figure here, we have the RNA being synthesized. So the DNA is shown in blue. The RNA that's being synthesized is shown in red. And the nucleotides are being attached to the 3' end, one at a time, pairing up with the complementary base pair using the DNA as a template. So the DNA is used as a template, so wherever there would be a T, an A would be added for the mRNA. Wherever there was an A U would be added. For a G, a C would be added, and so on and so forth, all being added to the 3' end as the RNA polymerase continues in this forward manner. And this is the elongation portion where it's elongating the mRNA by the addition of these nucleotides. And then we move into the termination of transcription. The mechanism of termination is going to be different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA at that point can be translated without further modification. In eukaryotes, the RNA polymerase II transcribes the polyadenylation signal sequence, and the RNA transcript is going to be released 10 to 35 nucleotides past this polyadenylation sequence. In eukaryotic cells, there is alteration of those mRNA ends. Each end of the pre-mRNA molecule is going to be modified in a particular way. The 5' end is going to receive a modified nucleotide 5' cap, and the 3' end is going to get what is called a poly A tail. These modifications share several functions. They seem to facilitate the export of mRNA to the cytoplasm, where it's going to eventually meet up with a ribosome. They protect the mRNA from hydrolytic enzymes so that it doesn't get degraded right away. And they help ribosomes in the cytoplasm attach to the 5' end of the mRNA. Here we can see an example of that mRNA with the addition of the 5' cap and that poly A tail. We can also see the polyadenylation sequence that was added to the end during mRNA synthesis. Now again, remember, these are modifications that are happening in eukaryotic cells. Not only do eukaryotic cells need alteration of these mRNA ends, but the genes also need to split where RNA is going to be spliced. So there are going to be two different regions that we see within the mRNA. Non-coding regions that are called introns, and other regions are called exons. Now these exons are eventually expressed. Usually they're translated into amino acids because these are going to be the coding regions of the RNA. So RNA splicing, after the different ends have been altered, there's going to be this RNA splicing, which is going to remove introns and join the exons. Now this creates an mRNA molecule that is ready to go to the ribosome because it is going to have a continuous coding sequence. So in a eukaryotic cell, all these extra steps are needed before the mRNA can be translated into protein. Again, we do not see this happening in bacteria. So here we have the pre-mRNA with its alteration of mRNA ends. Again, it has a 5' cap and a 3' poly A tail. It also has introns and exons within it. So what's going to happen is it has to be spliced where the introns will be removed and the exons will be placed together one after the other so that now mRNA can be read in a continuous coding sequence by the ribosome. At this point, the mRNA is ready to go to a ribosome. The removal of introns is accomplished by spliceosomes. Spliceosomes consist of a variety of proteins and several small RNAs that recognize the splice sites. The RNAs of the spliceosome also catalyze the splicing reaction. So the spliceosomes can come in to this area, remove the introns, and allow for the exons to be placed together. What is the functional and evolutionary importance of introns? Some introns contain sequences that regulate gene expression, and many affect gene products, even though they're not specifically placed into the gene product themselves. Some genes can encode more than one kind of polypeptide, depending on which segments are treated as exons during splicing. This is called alternative RNA splicing. And in alternative RNA splicing, there can be a number of different proteins that an organism can produce that is much greater than its number of genes. Because in alternative RNA splicing, we can sometimes take those different exons and put them together in different ways to make different genes. Proteins often have a modular architecture consisting of discrete regions that are called domains. In many cases, different exons code for the different domains in a protein. Exon shuffling may result in the evolution of new proteins by mixing and matching exons between different genes. So taking a look at this figure, we can see that in this case, we would have exon 1, exon 2, exon 3. The RNA would get processed to remove those introns and put those exons together. And this is an example of a protein having this modular architecture consisting of those different domains where each exon would code for a specific different domain of that protein. Now let's move into translation. In translation, the genetic information flows from mRNA to protein within this process. It is going to occur in the cytoplasm, and very specifically, it is going to occur on a ribosome. A cell is going to translate an mRNA message into protein with the help of another type of RNA called transfer RNA, abbreviated tRNA. And this tRNA is going to allow for the messenger RNA to be translated into protein. So we need the mRNA, we need this tRNA or transfer RNA, and then we need the ribosome, which is actually rRNA. So there are 3 different RNAs that are needed to assist or to be used basically to allow for mRNA to be converted into protein. So let's take an overview look of how this is going to happen. We have the mRNA, and then we have the ribosome, which is rRNA, and then we have the tRNAs, which are going to come in and recognize the different codons on the mRNA. Now on the tRNA, their codon is actually referred to as an anticodon because it is complementary to the mRNA codons. So it's going to be read in 3s, those codons on the mRNA. The tRNA will come in and recognize that codon with its anticodon, and based on the tRNA anticodon, it will be carrying a specific amino acid. It will recognize and bind to the codon of the mRNA, and it will deposit the amino acid to the growing polypeptide chain to allow for the mRNA to be translated into protein. So molecules of transfer RNA are not identical. Each is going to carry a specific amino acid on one end, and they're going to have an anticodon on the other end. The anticodon, as I just mentioned, is going to base pair with a complementary codon on the mRNA. Accurate translation is going to require two steps. First, a correct match between a tRNA and an amino acid needs to happen. So this is important because the tRNA has to then bring that correct amino acid into the ribosome to the mRNA to allow for protein production. But first, again, that tRNA has to bind to the correct amino acid, and this is done by an enzyme called aminoacyl tRNA synthetase. The second thing that has to happen is there has to be a correct match between the tRNA anticodon and an mRNA codon so that that amino acid gets placed where it needs to. What are the ribosomes going to do? The ribosomes are going to facilitate specific coupling of the tRNA anticodons with the mRNA codons in protein synthesis or translation. The two ribosomal subunits, large and small, are made of proteins and ribosomal RNA. A ribosome has 3 binding sites for tRNA. The P site is going to be the site that holds the tRNA that is carrying the growing polypeptide chain. The A site holds the tRNA that carries the next amino acid to be added to the chain, and the E site is the exit site where the discharged tRNAs leave the ribosome. So here we can see the 3 different sites on the ribosome, the A site, the P site, and the E site. So what's going to happen is the tRNA is going to go into the A site with the next amino acid that needs to come in. This tRNA is going to move to the P site, displacing the tRNA that was here to the E site, and it's going to leave. So in the E site, that tRNA is going to exit. This tRNA is going to bring the next amino acid in. It will move and add that amino acid to the growing polypeptide chain, which is going to eventually be the protein. Just like we saw in transcription, there are 3 stages of translation: the initiation stage, elongation, and termination. All 3 stages require protein factors that aid in the translation process. Energy is required for some steps as well. During elongation, amino acids are added one by one to the growing chain. Each addition involves proteins called elongation factors. Elongation occurs in 3 steps: codon recognition, peptide bond formation between the amino acids, and translocation. Energy expenditure occurs in the first and third steps. So there are 3 steps of elongation: the codon recognition, where the tRNA, the anticodon, is going to recognize the codon on the mRNA. That process requires energy in the form of GTP that is going to release energy for this process. And then we have peptide bond formation that is going to be the next step. And in peptide bond formation, that is where we're going to have one amino acid linked to the other, forming those covalent or peptide bonds between amino acids. And then finally, we have translocation, which is where the tRNA is going to move to another site on the ribosome, another ribosomal site, and this process of moving is going to also require energy in the form of GTP. Termination of translation is going to occur when a stop codon in the mRNA reaches the A site of the ribosome. The A site accepts a protein called a release factor. The release factor causes the addition of a water molecule instead of an amino acid. This reaction releases the polypeptide and the translation assembly comes apart. So again, here we see that the ribosome is going to reach a stop codon, which could be either UAG, UAA, or UGA on the mRNA. At this point, the A site is going to accept a release factor, and that release factor is going to promote hydrolysis, allowing the addition of a water molecule instead of an amino acid, and this frees the polypeptide and allows for the ribosome and other components to all dissociate. Often, translation is not sufficient to make a functional protein. Polypeptide chains are modified after translation or targeted to specific sites in the cell. During synthesis of the polypeptide chain, the chain is going to begin to coil and fold spontaneously into a specific shape. This is going to be a three-dimensional molecule with secondary and tertiary structure. A gene determines the primary structure, and the primary structure in turn determines the shape of the protein. Post-translational modifications may be required before the protein can begin doing its particular job in the cell. And these post-translational modifications occur after translation. So after translation, the protein has been made, there may be other things that need to happen to the protein before the protein is fully functional and can do its specific job. These are referred to as post-translational modifications. There are two populations of ribosomes that are evident in the cells. There are free ribosomes in the cytosol or the cytoplasm, and then those ribosomes that are bound to the rough endoplasmic reticulum. Free ribosomes are going to mostly synthesize proteins that function in the cytosol or the cytoplasm, while those ribosomes that are bound to the rough endoplasmic reticulum are going to make proteins of the endomembrane system and proteins that are secreted from the cell. So the endomembrane system includes the rough endoplasmic reticulum and the Golgi apparatus. Both of these things would be needed in order to secrete these proteins from the cell. Synthesis of these polypeptides always begins in the cytosol. So mRNA is going to attach to one of these free-floating ribosomes that are found within the cytosol. The synthesis is going to finish there in the cytosol unless the polypeptide signals the ribosome to attach to the endoplasmic reticulum. Polypeptides that are destined for the endoplasmic reticulum or for secretion are marked by a signal peptide. The signal peptide is a sequence of about 20 amino acids at or near the leading end of the polypeptide. A signal recognition particle, or SRP, binds to the signal peptide. The SRP escorts the ribosome to a receptor protein that is built into the endoplasmic reticulum membrane. The signal peptide is removed by an enzyme. Other kinds of signal peptides target polypeptides to go to other organelles. This figure shows an example of what would happen if there was a signal on the mRNA to take the ribosome over to the endoplasmic reticulum. So polypeptide synthesis is going to begin on the ribosome that is free floating within the cytoplasm. And then if there's a signal peptide that is there, the SRP is going to bind to the signal peptide, halting synthesis so that the SRP can bind to a protein at the pore, a receptor protein that is at the pore of the endoplasmic reticulum. The SRP will then detach and the polypeptide synthesis is going to resume now that the ribosome is attached to the endoplasmic reticulum. There's going to then be a signal cleaving enzyme that cuts off the signal peptide, and the completed polypeptide is going to fold into its final conformation now within the endoplasmic reticulum. Sometimes mutations can happen. Mutations are changes in the genetic material of a cell or a virus. Point mutations are changes in just one base pair of a gene. The change of a single nucleotide in a DNA template strand can lead to the production of an abnormal protein. If the mutation has an adverse effect on the phenotype, which is what we can see of the organism, the condition is referred to as a genetic disorder or hereditary disease. An example where we can have a point mutation that does allow for a phenotypic change is sickle cell anemia. So we can see here in hemoglobin what the wild type DNA would be in this specific area. And we have a T and an A in this area here. And it is going to switch to an A and a T. So the code is changing in this area. What's going to happen is what normally would happen, being read off the template strand, we would have an A on the mRNA here being a GAG in this codon. However, because of this change that happens in this mutation, we now have a uracil in the middle, making it GUG, and this is going to change the amino acid. And that amino acid is in such a specific place of the hemoglobin that instead of having a nice red blood cell that can carry oxygen the way that it should, it creates these sickled cell red blood cells that do not allow for oxygen to be carried appropriately. So this is an example of a single point mutation being in just the right spot that it causes a phenotypic change of this protein. It folds differently and it now can no longer function the way that it should. Point mutations within a gene can be divided into two general categories: nucleotide pair substitutions, or where there is one or more nucleotide pair insertions or deletions. So we can talk about substitutions first. In a nucleotide pair substitution, this is going to replace one nucleotide and its partner with another pair of nucleotides. Silent mutations themselves have no effect on the amino acid produced by a codon because of the redundancy in the genetic code. Because of that redundancy, because different codons can code for the same amino acid, those could lead to mutations that we don't see. Those are referred to as silent mutations. Two other substitutions we can have are missense mutations, which still code for an amino acid, but not the correct amino acid, and nonsense mutations, which change an amino acid codon into a stop codon. And what this means is that it stops the translation of the protein prematurely, which is going to lead to a non-functional protein. Besides substitutions, there can also be insertions and deletions. Insertions and deletions are additions or losses of nucleotide pairs in a gene. These mutations can have a disastrous effect on the resulting protein more often than substitutions do. Insertions or deletions of nucleotides may alter the reading frame. And remember, the reading frame is important, that the DNA and mRNA are read within the reading frame the way that it's supposed to be, because if the reading frame is altered, then it can produce a frame shift mutation, which changes the reading frame and would change the product. Spontaneous mutations can occur during DNA replication, recombination, or even DNA repair. Mutagens are physical or chemical agents that can cause mutations. Chemical mutagens fall into a variety of categories, and we will see that most carcinogens, which are cancer-causing chemicals, are mutagens, and most mutagens are carcinogenic. So there are different types of mutagens, physical or chemical agents that can cause mutations. So things like UV, X-ray, these types of things can cause mutagens. So things can happen spontaneously within the cell itself during DNA replication, recombination, or repair, or could be caused by mutagens, different chemical or physical agents that cause those mutations to the DNA.